Good morning, everybody. Whoa, little loud. Thanks so much for coming. For those of you who don't know me, Stefan Green from Rush University Medical Center. I'm the director of core lab services there. Really excited to have you all here. I came up with an idea for a session of like, hey, I just want to see more, like I always ask everybody, what's the coolest thing you saw? What's the new method that I should know about? And so I said, okay, let's just have a session on neat new stuff that is available to all of us as core facility directors that we can help our investigators get amazing data. So we've got 3 talks. I think we talked a little bit about it. We'll just power through the talks, and then we'll have plenty of time for discussion afterwards. But let me just introduce our speakers. Everybody can hear me okay, right? In the back, okay. So I'm going to give a talk. I'm a microbial ecologist, and so I always feel like microbiology gets a short shrift in new developments. So I want to highlight some of the methods that I think are super useful for microbiology. Also, I've got Alvaro Hernandez, who's the director of the DNA Services facility at the University of Illinois at Urbana-Champaign. He and I have worked together for, 15 years probably. And he's going to talk about some new epigenetic methods. And then we've got Alaina Weinheimer from the Bigelow Institute, who's going to talk about microbial single-cell genomics. So really kind of an exciting, hopefully, panel. And then once we're done talking, you can ask us questions, but you can also tell us, hey, you forgot about new methods that you should pay attention to. So, okay, let's get into it. I already gave you the session theme. Here's our talk titles. I'm going to channel my inner Chris Mason, for those of you who know, and try to get through this. I want to give the other speakers plenty of time. Okay, so a couple instruments and methods that I'm particularly interested in. Talk a little, like you've already seen a lot about N6. I don't want to belabor it too much, but we do have the ICON PCR that we've used. Also want to look at some new automation from a company called Volta with their Callisto instrument. And then also for some microbial ecology applications, I'm using a combination of both high-C and long-read PacBio data to do de novo bacterial genome assembly from environmental samples. And then also a cool company called Multitarga that is a startup looking at multi-amplicon reverse transcription. Then a sonication from a company called Triangle Biotech. And then I want to make a pitch for something I call medium read length sequencing. So not the short read that you know about and not the ultra long reads, but something in between, but at high throughput. Okay, I hope I don't have to tell you too much about the ICON PCR. Really neat instrument with a 96 well head. They now have a 16 well instrument. Each well is independently controlled, and it allows you cool options of for example, their auto-normalization, where you can amp to the same amount of fluorescence and then stop cycling on a well-by-well basis. The ICON PCR guys, the N6 guys, often focus on auto-normalization, balance your libraries, but from a microbial ecology application, we're also interested in reducing the total number of PCR cycles. So we've shifted a lot of our 16S ribosomal RNA gene amplicon sequencing to full-length 16S with PacBio, sequenced at UIUC. And one of the problems with long reads is that you increase your chance of formation of chimeras and other PCR artifacts that then eat up a lot of your sequencing capacity. So we're particularly interested. I always say PCR is your enemy. It's the greatest thing ever invented, but it's the enemy. So we're looking to use the adaptive thermal cycling from the ICON PCR to reduce total number of PCR cycles. Just showing you very little bit of data. We did a full-length PCR with 27F1492 primers. We have our own homebrew 2-stage PCR protocol. There's already a publication with it. We used 3 different types of samples, low biomass and low diversity skin, fecal samples and then high diversity soil. We did a temperature gradient, and then we compared between fixed cycles using 24 PCR cycles for the initial stage versus letting the thermocycler aim for a targeted amount of fluorescence and then stopping. So just, I don't want to, again, belabor it too much, but we have basically looking at our pass filter through our bioinformatics pipelines. We lose a lot of data when using PacBio relative to Illumina sequencing, but the losses are much better when you run fewer cycles. And so when we run the adaptive PCR cycling, we reduce from those 24 cycles somewhere between 5 and 9 cycles when using a targeted fluorescence threshold of 10,000. Of course, you can adjust that up or down to give yourself either more or less final PCR product. But the kind of fun thing is that we also, it's a little hard to see here, but even when we rarefy the data set to the same sequencing depth, we still see higher microbial richness with the fewer PCR cycles. So not only do you get more artifactual formation, it seems, but you also are selecting for dominant sequences in the PCR. And then finally, just made an ordination plot with different annealing temperatures and either icon PCR adaptive cycling or fixed cycling. And the fixed and adaptive cycling separate on the PC1 here, and then the annealing temperatures separate on the PC2. So you can see effects of the shift in the observed microbial community when you use the reduced number of PCR cycles. And I also always tell everybody, it's like the icon PCR is not like some weird instrument that came from nowhere. It's still a thermocycler, so you're still PCR cycling. You're just able to run adaptively fewer overall PCR cycles. Okay, also want to talk about the Volta Callisto. It's a low to medium throughput automation library preparation robot, but quite different. It uses electrowetting rather than standard liquid handling with pipetting. You can do, in addition to library preparation, Oxford Nanopore, PacBio, Illumina. You can also do high molecular weight DNA extraction. You can do hybridization capture. So just showing the kind of, it's a black box like everything else. But I got a video here. It's actually the first time I've ever used a video in a talk. So it's dispensing magnetic beads, and then it's able to vibrate them on the surface, and then it's got a magnet in there that it allows you to separate the bound nucleic acids, and then it's able to draw the liquid away. So you can do all sorts of neat protocols on there. It's very good, I think, for hybridization capture, where you can heat different liquids on the surface before you mix them. Okay, so using this, I just want to show just very briefly, we had an investigator who's looking to de novo assemble a bacterial symbiont from an insect. He sent us genomic DNA. We sheared the DNA using G-tubes, and then made PacBio libraries on the Volta. We sent it down to UIUC. Actually, they had a failed run the first time, and then reloaded with just a little bit of library that we had left over. Sequencing still went great, and we were easily able to de novo assemble the bacterial symbiont from the long inserts. So the libraries look very good that come off the Volta. I also want to talk just very briefly about a combination of two methods that have been around for a long time. Of course, you can do shotgun metagenomics with PacBio, get long inserts. And Hi-C also has been around for a while, and we put them together for a project that I'm working on. It's called the MHU-8 mission. It's a NASA mission to look at, and my part, to look at the effects of spaceflight on the gut microbiome. So one of the really cool things about the MHU-8, it's the Mouse Habitat Unit 8 mission. It's a partnership with JAXA Labs from Japan. They've got centrifuge cages up on the International Space Station, and you can spin them at different rates to generate artificial gravity in space. So you can start to separate out what are the effects of space flight. Is it microgravity? Is it diet changes? Is it radiation? And so we have 4 different treatment groups at microgravity, 1/3 Earth gravity, 2/3 Earth gravity, full gravity. 6 mice per group. They're in these little cages, individually housed. And one of the things we found is when you individually house animals, you get much less difference between ground and flight. We don't think there's a huge effect of space flight on the gut microbiome. But we did find one organism that has a nice dose response to gravity. And so then we went back to the fecal pellets that had the highest level of that, or that animal had the highest level of the organism that we were interested in. And we did a high C on that, and then also extracted DNA, sent for PacBio, again down at UIUC. And then using a high C coupled with, and sorry, I should just say all of this is from Adith Chowdhury, who's the bioinformatician on this project. And using those, we were able to assemble the genome into 2 contigs. The closest relative from pure culture in NCBI is 250 contigs from a pure culture, and we're able from this environmental sample get 99% of the genome, and probably we could finish it with Sanger sequencing. So I think in combination, really a very valuable microbial tool, a little cost dollar intensive, but hopefully we'll see drops in pricing there. Sorry to just come at you, but like I said, want to make sure everybody's got time. So I'm also really interested in Multi-Targa, a company that's building a custom multi-amplicon panels for reverse transcription that then will feed into RNA sequencing workflows. Okay, so the idea is that instead of having to do ribosomal RNA depletion or any other enrichment strategy, you can, for example, again, coming in from a microbial perspective, if let's say you're looking at lung tissue and you're interested in Pseudomonas aeruginosa, you can design reverse transcription primers for every gene in Pseudomonas and use that pool in place of random primers in your RNA-seq workflow. So kind of like, for me, kind of an amazing idea, because then, for us in the microbial world, the ribosomal RNA depletion and the lack of polyadenylation in the messenger RNAs is the nightmare that makes you wish you worked on eukaryotic organisms. So this is another strategy where if you know what the organisms you're looking for, then you can just a couple bucks in primers, and then you're able to skip a ribosomal RNA depletion stage. So it's not just for microorganisms. So you can also do targeted oncology panels, just looking at reverse transcription of those genes that you're interested in. So kind of high hopes, and you can also deselect for the organisms that you don't want in the design. Finally, in terms of kind of other interesting things, I work with a company, Triangle Biotechnology, that has a reagent that you can add into your library or nucleic acids that you can sonicate, but you don't need a kind of super high energy sonicator like a Covaris. There are these lipid droplets that kind of explode when they're hit with sonic energy, and allow you to generate that same cavitation energy to do DNA shearing. But the kind of cool thing is you can also attach all sorts of things to those lipid droplets, including antibodies. So you can also try to selectively lyse cells by having the antibody grab onto a cell. Then you hit it with sonic energy, and it's right next to either the cell wall or the cell membrane. So you can enhance cell lysis that way. But just they have also come out with a 96-well sonicator that can be emplaced on an automation deck as well. So if any of you want to skip over doing enzymatic lysis or enzymatic shearing, you can use this device in place. Okay, last thing I kind of want to mention, like I said, I want to make a pitch particularly for us microbiologists, for that medium read length sequencing at scale. Okay, so bacterial genes are often on the scale of 1,500 bases. We don't have, for the most part, introns. So 1,500 base reads will get you, let's say for shotgun metagenomics, close to full-length gene that can be annotated really well without de novo assembly. But we need it at, billion sequence scale. I think also if we can get even a little bit larger than that, maybe 3,000 bases, 3,500 bases, we can do de novo assembly of bacterial genomes without any long read or high C technology. So push your sequencing platforms to don't forget about us. I think the closest platform right now seems to be Illumina with their 2x300 on the NovaSeq X, but also Complete Genomics has a 1x600, and they're talking about getting a 1x1,000 at the billion or 2 billion cluster scale. Maybe the Roche platform eventually will be able to do it as well, but I just think there's huge value in that intermediate read length that we're neglecting. So, okay, thanks everybody, and I'm going to hand it over to Alvaro. Oh, not bad.
Okay, so thank you, Stefan, for inviting me and everyone for coming. I'm Alvaro Hernandez. I'm the director of the DNA sequencing facility at the University of Illinois. Back in November, Stefan asked me, hey, I need a title for your presentation. And I asked Copilot, hey, I need a title for my presentation. And this is what came out, but actually today I'm going to talk about all the epigenomics methods that we use in the lab or that we are planning to use. First, I'm going to introduce my lab so that you guys know what we do. We are a center at the University of Illinois. We have a centralized model in which several core facilities are under the umbrella of the Roy Carver Biotechnology Center. So we have DNA services and functional genomics. The director of the functional genomics, he just retired, so we are now merging it with my facility. We also have bioinformatics. We have flow cytometry and a proteomics and a metabolomics facility. The DNA services facility is the largest one. This is an old picture. We are now 14 people, and we are fully dedicated to service. We bring the technologies that we think are the best fit for the needs of our campus, and we also offer end-to-end service, meaning we go from consultation on experimental design, how to collect the samples. Then we also do DNA and RNA extraction. We make libraries. We do the sequencing and then the analysis. Like many cores here, we get samples from all over the United States and from many other countries. So regarding the instrumentation, our short-read technologies are the NovaSeq X Plus. This is the workhorse for everything we do that needs billions of reads. And we also have two MySeq i100, and we are in the process of getting another NovaSeq and another MySeq. Regarding long reads, we mostly use the PacBio. We use it to do a lot of de novo genome assembly, to sequence full-length 16S metagenomes, and we also have the Oxford Nanopore GridION. We offer the entire suite of single-cell and spatial transcriptomics. We are mostly at the Next Genomics shop, so we do single-cell RNA-seq, ATAC-seq, multiomics, and we have a Visium. We offer the Visium HD and the Xenium. We still have a lot of demand for microarrays, so we mostly have demand for methylation microarrays, and we also offer the SNP microarrays, mostly for ancestry studies. And Sanger sequencing, still here. We have two of these 3730X cells that are still really busy. Not everybody needs to look at the entire genome. So now we are going to talk about epigenomics, and research in genomics involves either looking at the genome, whether you are doing de novo genome assembly, or you are doing, looking for genomic variants, or looking at the transcriptome, or looking at the epigenome. And today I'll talk about the epigenome, and so we are all on the same page. Epigenomics is the study of all the modifications of the genome that regulate gene expression without changing the sequence of the DNA, right? And so these modifications could involve, for example, how the chromatin is folded inside the nuclei, what parts of the chromatin are accessible, are available for gene expression, what type of methylation is there in the genome, also, what transcription factors, where do they bind, what histone post-translational modifications are there, and also RNA modifications. So the epigenomic mechanisms that I'm going to talk about, I divided it in two. One is when looking at the chromatin, the DNA is inside the nuclei, is all folded. And for those, we have looked at, we have used 3D genomics. We also look at chromatin availability with both in bulk and in single cells. I'm also going to talk about doing chromatin availability and methylation at once in the same library, so that's the technique called fiber-seq. And then I'll talk about transcription, doing characterization of transcription factors and histone methylations. Then when we extract the DNA, we remove all the nucleosomes. Now we have a relaxed DNA strands. So then we can look at whole genome methylation using EM-seq, or looking at the 5-base techniques, or looking at the 6-base genome. And I'll also talk about reduced representation libraries. So regarding 3D genomics, and this is the DNA inside the nucleus is completely folded, and there are several topological features in the DNA. One of them is the AB compartments, and the AB compartments means that there are regions of open chromatin. They kind of like to be together in their own parts of the nuclei, and portions of closed chromatin that like to be by themselves. So those are the AB compartments. So the regions that have mostly open chromatin are the A compartments, and regions that have mostly closed chromatin are the B compartments. When you look at the DNA strands, these are regions 100 kb to megabase size. There are some points of contact, preferential points of contact. So there are points where the DNA strands touch each other, and these seem to be quite stable. So these are called TADs. These features are called TADs, and they are very stable. They are very stable in different cell types and also across different conditions. But if you look inside the TADs have a lot of loops. So loops is a very defined feature of the DNA strand, and they also have some preferential points of contacts. But the loops are very dynamic. They change on different conditions and different cell types. So what happened was that a PI told us last year, about August or so, that they had $70,000 that they had to spend before the end of the month because of a grant that expired, and that he wanted to do 3D genomics, which we had never done before. I looked in Google, and I found that Dovetail Genomics was. I'm going to name several companies. None of them pay me, or ask me to name their products. I'm just saying these are the kits that we used, right? But I saw that Dovetail Genomics had a kit for 3D genomics. We called them. We had a webinar. They told us how to make the libraries. And the PI was in Chicago, so they had to send us the tissues. They collected brain tissues and brain sections, and they froze them, and they sent them to us. Then the next step is to do a cross-linking to preserve those contact points in the DNA. And then the assay is based on transposition. So it's a really quick assay. You do a transposition, you make the library, and then you sequence it. Sequencing depends on what you want to look in that chromatin folding. If you are just interested in AB compartments, then you can sequence it to a 10x depth. Just one library, 100 million reads per library. If you want to look at TADs, then you have to sequence it to 30x, and loops, because they are a much more fine scale conformation. Then it needs 3 libraries and 800 million reads in total for the 3 libraries. So the libraries can be sequenced for up to 300 million reads per library. So it was really easy, really quick, and now we have experience in 3D genomics. The other technique that probably a lot of courses do here is ATAC-seq, right, where we are mapping the portions of the chromatin that are open. And ATAC-seq is based on transposition, so the DNA that is wrapped around the nucleosomes is protected from transposition, so transposase only gets inserted in the open chromatin. And so ATAC needs, we usually do use 20,000 to 100,000 cells per sample. It needs high cell viability, because if a lot of the cells are dead, then they lose the chromatin structure, so you end up making very expensive shotgun libraries. Initially, the first protocol that was released did the transposition in cells, but then a very high % of the reads were for mitochondria. So now there is a second iteration of protocol that perform a nuclei isolation, a very crude nuclei isolation, and then the transposition, and that reduces the amount of mitochondrial reads. It can be done in frozen tissues. Usually, when people tell us, can I freeze my tissues? I tell them to call the companies to tell them, how to do it properly, and we always test if it worked or not. So they tell them to send us test samples. We make a library, and we look at the traces of the library. Sometimes it works, sometimes it doesn't. And this ATAC-seq need 50 million read pairs per libraries. And this is the, I don't know if I can see the point. No, I cannot see the pointer there. But this is the huh? Oh, there you go. Awesome. There you go. Excellent, perfect. So these are the traces of the libraries. And here, you can look at these black lines in here. ATAC-seq libraries have this periodicity in which the first peak comes from a transposition that happened in open chromatin, so no nucleosome. It's a nucleosome-free region. The second one is the cut happening between one nucleosome or two nucleosomes or three nucleosomes. So what we want is most of the reads coming in a nucleosome-free fraction. So here the blue trace is also a very good one, mostly nucleosome-free, and some mononucleosome and dinucleosomes. The green line, the green trace is okay. It's kind of it's missing the periodicity, but it might be still okay. But the red tracing here, that's a shotgun library. So something happened. A lot of cells were dead. A lot of nuclei were, disintegrated. So there was no chromatin structure. So we do this in bulk and also single cell ATAC and single cell multi-ohm when we do single cell RNA-seq and ATAC-seq in the same nuclei. But the reason why I'm talking about ATAC-seq is because of the next technique. This is fiber-seq. This is the only technique that allows you to look at both open chromatin and methylation. And the way they do this by using an enzyme that methylates accessible A's, so adenines. So any A that is in an open region of chromatin gets methylated. Then you extract high molecular weight DNA and sequence it with PacBio. And what happens is that PacBio can natively recognize methylated Cs, and also it can natively recognize M6A. So you will see these clusters of methylated A's. Those are coming from regions that were open chromatin. So this is open chromatin, and it will also recognize, of course, the 5MCs. Right now, this is a very expensive technique because you can only sequence one sample per run. But PacBio is about to release these reusable flow cells, and that's when I hope that we will be able to sequence at least 2, but probably 3 samples in one run. And also they are about to release a new software that allows for the recognition of 5MC and 5HMC. So I think we will be able to, hopefully we'll be able to look at chromatin accessibility, 5MC and 5HMC in the same sample. And this is the only technique that allows you to look at these 2 epigenomics markers. The other technique that we use a lot is cut and run. So when I started working with Illumina, that was back in 2008, I had one of the first genome analyzers, and all I did was ChIP-seq for several months. ChIP-seq, is based on shearing the chromatin and then using antibodies that recognize a transcription factor or a histone modification. But ChIP-seq has, a lot of non-specific background, right? So you pull down your specific antibody, but also there is quite a bit of background. So ChIP-seq needs quite a bit of sequencing, so 40 to 60 million reads per sample to differentiate specific peaks from background. Cut and run is a newer technique that instead of using shearing of the chromatin, you do the antibody, an antibody-based recognition of the protein of interest, and then you use a micrococcal nuclease to cut around the protein. So it's much more specific. You only need 5 to 10 million reads per sample, so also way cheaper. And there are two techniques. One is cut and run, the other is cut and tag. Cut and tag is based on transposition, so it's much faster, but it's only good for histone modifications, whereas cut and run is much more robust and is for all proteins and for all cell types. The traces of the cut and run libraries are similar to the ones from the ATAC. You have to see this periodicity here. So that's about epigenomics mechanisms in chromatin, but now let's purify the DNA, get rid of the nucleosomes, and see what we can do with purified DNA. And the technique, what we do the most in our lab is we do EM-seq. So we are looking at methylated cis. And for that, we use the EM-seq kit from NEB, which it has first a reaction that protects the methylated cis, and then the next reaction is deamination, which converts all the non-methylated cis to Ts. So basically, 97% of the Cs in the genome are non-methylated. So you convert all the non-methylated Cs to Ts, and all the ones that remain as C are the methylated ones. For this, we usually sequence 50 libraries per 25B flow cell. So that gives us a 30x depth of the genome, and so we target about 550 million reads per library. And we do the analysis of the EM-seq libraries in the NovaSeq using Dragon. And this is a huge value to our customers because, first of all, analyzing 50 libraries in Dragon takes 12 hours overnight. So when I come to the lab, the analysis is done. And my customers tell me that doing this even in a high computer cluster, it takes them 1 day per sample. So it takes them a month and a half to analyze the libraries, and I can do it in the NovaSeq overnight. They also have, so NEB now has a separate module that to make a second set of libraries to detect 5-HMC. But it is a second set of libraries that needs a separate sequencing. Now, going from this, now Illumina released the 5-base genome, and Watchmaker also has one. They just talked about it. This is a complete change in the way of doing methylation, because instead of converting the non-methylated cis to Ts, you convert only the methylated cis to Ts. And this is major because the majority of the cis are not methylated. So by only converting the methylated cis to Ts, you leave the rest of the genome intact. Okay? So now you can do, in the same library, in the same sequencing, you can do both detect methylation and also detect genomic variants. Okay, so this is really something very new. And Illumina released this kit in January. We already had a project waiting for it. This one, the recommendation is to sequence at least 30x, but mostly 40x. 30x would be just for methylation, but you need a 40x for the genomic variants. And if you're doing normal tumor, you can also do enrichment using probes, and you can then sequence the tumor at much higher depth. So we did 40 libraries in a 25B flow cell, and Illumina has an app to do the analysis in base space. And I don't remember if you pay $5 or $20 per sample, something like that, but and it's also super fast, and you get all the genomic variants, SNPs and insertions and deletions and copy number variants, and the methylation also. The next application is the 6-base genome. This is from Biomodal, and what they do is the following. They produce a library in which read2 is a synthetic copy of read1. And they create some pairings so that if in read 1 you have an A and in read 2 you have a T, A and T, that will call an A. And if you have a T in read 1 and an A in read 2, that's a T, and so on and so forth. And here, for example, if you have a C and a G and a G and a T, that's 5-hydroxymethyl cytosine. So it's one library, and you get both you get genome, 5MC, and 5HMC. It is a very long protocol. It takes like 3 days, and you need a very high sequencing depth, so about 1 billion read pairs per sample. We still, not everybody, sequencing entire, doing methylation in the entire genome is still, expensive depending on how many samples you have. So we still do a lot of reduced representation libraries, which was the first method that was available for whole genome methylation. And this one is based on first digesting the DNA with MSP1. MSP1 cuts mostly in CPG islands. And so it creates fragments 40 bases to 300 bases in length, and you get mostly methylation in promoter regions. But you can sequence 20 libraries in one 10B lane, or 60 libraries in one 25B lane. So it does get really, really cheap. It takes about 3 to 5 million methylated Cs out of the total of maybe 30 million or so. So it's a very low cost to characterize differences in methylation patterns among different samples. And at the end, just a little bit about RNA methylation. This is what we use the most. Our customers actually made these libraries, the enriching RNA fragments that have M6A methylation. So this is basically like ChIP-seq, but in RNA. These libraries need a lot of RNA, like 10 micrograms of total RNA. There's a kit from Epigenetech, and it needs some validation, let's say, to get it to work. But once they get it to work, it works, let's say. And in each library, you have to sequence 10 million reads. There is now a new kit from Alida Bio that will enrich for both M6A and inosine. And the advantage of this kit is that it only needs 250 nanograms of total RNA instead of 10 micrograms of total RNA. And also, I like the fact that it has in vitro controls. So it has a control, so you can see the efficiency of pull down and the precision, in calling M6A or inosine. And it has UMI, so you can remove all the PCR duplicates. And it allows you to do RNA-seq quantitation of gene expression and methylation at the same time. And the next one is Oxford Nanopore. This one, with direct RNA, they say that you can, detect all these methylations. The problem right now is that they don't have barcodes, and so you can sequence only one sample per flow cell. They say that now in April they will release barcodes, so that will make it cheaper to do. And one thing that I would recommend when doing Oxford Nanopore is to generate your synthetic controls, because otherwise I don't know if they are calling the right methylation and the efficiency of this calling. We are planning to do it, but we will definitely generate our own controls. And with that, thank you very much for listening.
I don't know how, a little shorter, sorry. Hi, everyone. I'm Alaina Weinheimer. I work at the Single Cell Genomics Center at the Bigelow Lab for Ocean Sciences. Just a little introduction of what that means. The Bigelow Lab is a nonprofit research institute, but we also have services. And so the Single Cell Genomics Center is like part of its role is providing services for internal and external customers, but then we also do technology development research and ocean research. I'm kind of in the ocean research arm that got pulled into some technology research. And so what I'm presenting today is one of the latest technology developments coming out of the lab. And I know it's a long title, so this is the title from the publication. Well, kind of. So I just like renamed it Ramping Up Single Cell Genomics with Bubbles. That's a little more, says the same thing. So kind of one of those fundamental goals of microbiology is to be able to know who's in your sample. And we've heard a lot of different ways to look at that. But right now, the common way is, shotgun metagenomics, where you take, or bulk sequencing, where you take all the DNA from your sample and then you chop it up for whatever read length you're going for, and then you assemble that back together into metagenome assembled genomes with computational tools. And these tools are really good at assembling genomes that tend to be less complex, or maybe rare distinct members that don't have as much microdiversity that could break your assembly. And there's been benchmarking work on that. But I liken this concept to, taking a lot of different puzzle pieces together, mixing them all up, and you tend to get back puzzles that are easy to put together, or, pieces of maybe rare or more distinct things. So an alternative to this is to just separate all the individuals and then sequence their genomes from there. And so that's what the Single Amplified Genomics Center, or Single Cell Genomics Center, focuses on. We right now use fluorescence activated cell sorting. Sorry, I'm like confused of what I'm seeing on the screen. Where we use flow cytometry, we pass it through different channels until the sample is so narrow that each particle is lined up individually, and then we shoot lasers to detect whether there's DNA, and then we use the backscatter to see if it's the size of what we're targeting. So if we're targeting cells, is it the size of a typical microbial cell, or is it the size of a virus if we're targeting viruses? There's not enough DNA in a single cell, so we always have to do a genome amplification, which is not ideal, but that's not enough DNA in these guys yet. So then we can prepare a library, chop that DNA up, assemble it all together, and we get a single amplified genome, or a SAG. What this method ends up capturing are genomes of abundant members, because the cells or viruses are randomly sampled, and you're getting lots of copies so you're getting that individual resolution of genomes with this kind of approach. However, the throughput is kind of low. So a typical plate, we have 384 well plates, and we have controls on there, so we tend to get less than 384 genomes per run. And then there are some optical biases, because we're looking at size to determine whether or not we sort that particle. So some small viruses end up looking like noise in our flow cytometer, and then conversely, there can be small cells that get discarded as viruses, and large viruses that aren't cells, and so on. So there's some challenges there. So the motivation of this work was to find a new way to separate individuals. And so for this, we partnered with Trendy Biosciences, which is a biotech company based out of Vilnius, Lithuania. I don't know if you've heard of Droplet Genomics. They're now a part of Trendy Biosciences. Am I picking up, or am I too close? My volume's okay. Okay. So they developed this technology called semi-permeable encapsulation. And what that means is it can capture individual cells or larger particles, long stretches of DNA or viruses, but then it's permeable, so you can have enzymes and reagents flow in, and you can do multi-step reactions this way. So to show you what this capturing of particles looks like in the lab, it's all done on these desktop flow cells. Some of them are even this small. The one we work with is a little tall. But it's tiny. It's not this massive flow cytometer and that we typically use. And you can see here, this is a video camera of what's going on top of this instrument, where we have our samples streaming through one channel and our reagent to make those capsules streaming through the other. And I hope this video works. You guys see that? It's really hard to see. I'm sorry. But basically, we're forming little bubbles as our sample's going through, and about hundreds of thousands are formed in a run. And so when you combine this with single-cell genomics and using our whole genome amplification and everything else, we call this method environmental microcompartment genomics. If you have other names for ideas for names, please let me know. It's very long to say. But we are showing this pilot application on seawater because we're an ocean lab. But you could imagine this for any type of environment, from human gut material to sediment and soil, wastewater, whatever you're interested in. And at least for our pilot application, we didn't do any pre-treatment. We just took a seawater sample, which has bacteria and viruses and free-floating DNA and vesicle-captured DNA, and then we compartmentalized it into these semi-permeable capsules. They're about 70 micron in length. Some of them are smaller now, but we dilute the sample first so that we don't end up with cases where there are multiple bacteria getting captured in one of these capsules because they're the width of them. And then we can perform that genome amplification, and then we do combinatorial barcoding, so that we can label all the DNA inside that individual particle with its own barcode. And then we can pop all those bubbles and pool all that DNA together, sequence it, and assemble it, and get lots of single amplified genomes. And so I'm going to be showing you the results from our pilot application to seawater. So we collected this seawater off the dock of the Bigelow Lab in Maine. We're located on the coast of Maine, about an hour north of Portland. And we took this 300 nanoliter sample, so 0.3 microliters. We didn't do anything to it because it's so small. And from that, we got over 2,000 SAGs. So the efficiency of this was pretty high based on what we expect from microscopy. Because the one limitation of this method is that we don't have optical properties. So we don't know if that genome's coming from an intact cell or a vesicle, or if it's free-floating DNA. And so our first task when we got those genomes was to determine whether that genome is cell-like or virus-like. And virus-like, think mobile element-like, non-cell-like is maybe a better word. And we did it based on some properties in their genome. I can elaborate on that later. But when we tallied up the number of genomes belonging to these categories, about 88% of our genomes were virus-like, and 9% were cell-like, and then 3% we couldn't tell which. This ratio of viruses to cells actually matches what we see when we use microscopy and other methods on seawater, so this was really reassuring. And my advisor was super happy because my background is in viral genomics, and he's a cell genomics guy, so this was a great meeting of minds for this kind of data. So since we mostly got viruses, we wanted to see how the community from our 300 nanoliters in this new method compared to other methods. So we had like a big vat of seawater where we took 300 nanoliters, and then we also performed other methods on that same vat of seawater. So I'm going to be showing you some genome quality results. So our EMG-CG SAGs, we had about almost 2,000 viral genomes. We also performed fluorescence-activated virus sorting, so using that flow cytometer, and we targeted virus-sized particles, which we only got 190 genomes from. It's really hard to sort viruses from seawater, at least. And then we also prepared a virus-enriched metagenome where we filtered out the cells and then just sequenced what the filtrate. And we got about 17,000 mags from 40 liters of seawater. That sounds like a lot of genomes from the metagenomic approach. However, we then looked at the genome quality of what we recovered. And so we used a tool called CheckV. I don't know if CheckM, it's kind of similar to that. The category I'm focusing on here is medium. It means it's 50% complete or higher. And as you can see, over half of the genomes from our EMCG SAGs were at least 50% complete or higher. Whereas for the MAGs, they were less than 1% had medium completeness. When we then clustered these genomes together to see if we're recovering the same viruses or not, we often saw into species units. We often saw cases where there'd be one SAG with several contigs and then several different MAGs aligning to different parts of that genome, showing how fragmented MAGs can really be, at least for viruses in complex environments. And right now in viral ecology, we usually refer to a MAG or a VOTU as its own species. So if you were to look at this sample, you'd think there were 14 different species there, but they actually all belong to the same species. So it's just interesting to see how these different methods give you different metrics. But we only sequenced 300 nanoliters, and the ocean is a lot larger than that. And so there is still a lot of value in the data from these different methods. And so we used the reads from the metagenome to look at the abundance of our virus species in 40 liters versus 300 nanoliters. And so here I'm going to be showing you a plot where each species on the x-axis, their abundance in the 300 nanoliters is the number of genomes we recovered from that species. Because the beautiful thing of SAGs is each genome is an individual particle in the environment, and it's randomly sampled, so it's somewhat quantitative. And then for the metagenome, we took the reads that mapped to that genome and averaged it for all the members of that species. I'm sorry, I have so many animations. And then compared those reads to estimate their abundance in the 40 liters versus the 300 nanoliters. And we expected a positive correlation because the ocean is mixed. And so things abundant, would be abundant in a small bit versus a big bit. And we did see a significant positive correlation. However, as you can see, there's a lot of variation down at the more rare things. But what's most striking to us, though, was that our most abundant species was undetected in the metagenome. And we first thought, okay, is this something strange? Is something wrong with our method? What's going on there? And so we then looked at relatives of what these viruses were, and they belonged to the novavirus genus. This group of viruses was only discovered recently, and some of their biology makes a lot of sense. So I'm just going to briefly describe novaviruses. So this is a typical phage. This is a picture of, there's only 2 of these guys in culture. They were isolated 5 years ago on the bacteria Rosiobacter. This one comes from the Grand Venice Canal. The other one comes from the coast of Mexico. And it looks like your typical phage. But when the researchers went to sequence its genome with Illumina, just direct sequencing, they couldn't get a genome. And so they then tried, and they actually contacted New England Biolab about how to go about getting this genome. And they found that they were able to get a genome after they did whole genome amplification. And it turns out that these phages actually have non-canonical DNA. So instead of having a deoxyribothiamine in their genome, they have a deoxyribonuridine. So it's just lacking that methyl group. It's a precursor of thymine. So it's there, but these guys actually incorporate it into their genome so significantly that you can't just sequence it and assemble, and you can't detect it in metagenomes. And so the reason that they were able to get this genome after amplification is because the polymerase used to make the genome copies accidentally reads that uridine as a thymine. So then Illumina can then sequence the copies of that genome. And so, like our pipeline, we also use Phi29 polymerase to make copies of our genome. And so it made a lot of sense why we're able to find these viruses because we also use that genome or that polymerase. So what's most striking about this is that, this species was the most abundant species in our 300 nanoliters. We typically in viral ecology, we use metagenomics, and we have not been able to find this virus before. And so it makes me wonder if the canonical is non-canonical in the virus world. What is canonical? The other cool thing about single cell genomics or single amplified genomics is that we can use cell-sorted SAGs to link viruses with hosts. So when you're sorting a cell in using the flow cytometry approach, if a cell is sorted and it's infected by a virus at the time of sorting, you will sequence both the virus and the cell genome and the viral genome in the same sequence genome, depending on the stage of infection. And so we then went back and looked at previously sequenced cell genomes from the Gulf of Maine, and we were able to link these viruses with some new hosts. The quality, I am so sorry, I didn't realize it was that poor, but we were able to link these viruses with Amylobacter, which is a relative of the host that was cultured, but then also Pelagibacter and 2 Pelagibacter cells. And Pelagibacter are the most abundant bacteria in the ocean. So it's pretty shocking to see that not only were these viruses the most abundant species in our sample, but they also might be infecting the most abundant bacteria in our sample. It makes you wonder what else is out there in your system that you just couldn't sequence before. And so that's it, all I have, and I'm going a little over time. But if you're interested in hearing more about this method and different parts of the pipeline, because you can buy this equipment to make the capsules yourself, but we also offer the bioinformatics and support and so on. You can check out our center. The center QR code is there. There's a picture of some of us down here. And I'll just leave up my conclusions and acknowledgements. And yeah, the paper's in Nature Microbiology if you want to check it out. Thank you.
All right, great. So thanks to all of our speakers. I think we've covered a broad range of different methods and instrumentation. I just want to reiterate, I don't think any of us have received any money from any of the companies. So this is, and particularly on my side, I don't mean to disrespect any company that I didn't cover. There are many more methods and instrumentation than we used. But I'd really like to open it up to the audience now. Any questions for us, of course, but also if you want to highlight for everybody else in the group something that we missed or other really impressive advance, we'll be delighted to hear from all of you. Oh, and I guess we've got a microphone set up.
Can I bring this to you?
Just regarding the last topic, what's your success with long-read sequencing?
Oh, yes. So unfortunately, the amplification step, and I was just discussing this last night, it can, like the way it's amplified can actually create chimeras. And so if we end up doing long read, the fragments that are amplified are actually not representing the conformation in the genome. So short read actually helps us avoid getting chimeras, ironically. So unless the amplification step can change or we can fix that, then we can't do long read yet. Yes, we're sequencing individual cells. So even if we have lots of cells, we're still working with whatever DNA is in one cell.
Have you tried PTA from BioScribe, where you don't get the same kind of explosive amplification? There's terminator bases in there, so you get smaller inserts than the whole genome amplification. You might try that.
Yeah.
You probably could play around with the terminator, like the ratio of terminators, to see if you can get longer fragments. I think they top out around 1,500, 2,000 bases. But it might still be better than what you have now.
Thank you so much. I have a list of new cool toys that I'm going to Google right after this. Well, right after I went to see the vendors. But just update, you probably are aware of this because you have talked with Alaina Bio, but they are adding pseudouridine to their kit. So then you're going to have like the gamut of major RNA modifications. I don't know when it's coming. I had a chat with them recently because I'm also looking to add that to the lab.
Yeah.
So I'm really happy to see that you are doing it too. I may
Definitely.
ask you questions.
They say they are offering it in-house, but it's not available commercially yet. But we are also looking forward to it. Yep.
All right, I have a quick question about you on your journey to medium read sequencing lengths. Have you tried pushing some of the 2x300 kits, particularly with Element as a 1x600, or even reading longer on some of those existing flow cells?
I haven't really played with it. Alvaro is my primary sequencing partner, so we can talk about it. We have, of course, got the I100, which now has a 2x500 chemistry, which we've started using for 16S amplicon sequencing. So just let everybody know, you can actually load a 1500 base pair library on the I100. Of course, the reads aren't going to overlap, but you can concatenate the forward and reverse read, for example, and use annotation that way. So there are already possibilities to get long inserts. I talked to Illumina. They said you probably can go maybe 700 bases, 750 in one direction. That's great. The problem is that the flow cell that the 1,000 cycle chemistry is running on is only the 25 million cluster flow cell. So, if you've got a metagenomics project with 500 samples, it's not going to work out. So that's why I really want that at scale, but medium read. And I just feel like it's been neglected. Illumina took a long time to get to say, okay, hey, we can't just push the total output, we need to also push the read lengths. And I'm hoping we can jumpstart Roche to start thinking not just about, okay, we're going to get 80 billion reads out of this thing. I'm like, okay, give me 10 billion reads, but longer.
Yeah, it's a real Goldilocks zone between a missing gap in read length and then looking at getting a reasonable output. So thanks for sharing that. Who else wants the mic here? There was good stuff. No good questions?
Come on, guys.
I actually have a question also. I have a question for Stefan about the icon N6. We were always told, for example, when making RNA-seq libraries, that all the libraries have to be started with the same amount of RNA, they have to be processed the same way, they have to have the same number of PCR cycles, that the only difference between the RNA-seq library should be biological and not technical. And same thing with 16S. But now with the icon, we are being told that differential amplification is okay. So can you comment on that? I mean, is it okay to amplify different RNA-seq libraries with different cycles on the same project?
I think it's one of the greatest questions I heard. I mean, it's really, really important. I think, at least from my perspective, when you put in the same amount of, and again, I come from a microbial perspective and not so much on eukaryotic. So when you put in, I don't know, 5, 10 nanograms into a reaction, let's say even a PCR reaction, that doesn't mean the same thing for every sample. So if you put in 10 nanograms of fecal DNA that you're PCRing from one sample versus another, the percent host DNA that's in there, the percent off-target DNA that's in there, actually means you have different numbers of copies of targeted template, for example, in a PCR reaction. So 5 nanograms doesn't always mean 5 nanograms in terms of amplifiable target. And so I think there, the icon PCR can really help, because then again, from my perspective, you're always trying to limit the total number of PCR cycles to avoid the bias that are introduced. It's worse, I think, for microbial, 16S amplicon sequencing, where you have primers that are heavily degenerate. So this is an area of obsession with me. But you've got degenerate primers that are causing bias, and the more cycles you run, the more bias you introduce. So from that perspective, both from the 5 nanograms doesn't mean 5 nanograms. I think the icon PCR should work perfectly. I don't think we've seen exactly the data whether, if you're doing eukaryotic RNA-seq and they're all RINs of 10, should you normalize the RNA going in, or should you just let icon PCR normalize it? I think that's maybe still yet to be determined. But you might talk more with the N6 guys to see if they've done that. We haven't. We're not super careful usually on precisely 5 nanograms or 10 nanograms. Often we have investigators who are like, this is all the RNA I've got, so it's usually craptacular and all over the place in terms of total amount of RNA. So we don't have the luxury of always putting in the same amount of RNA for our samples. I don't know if it's just me. We get more crap samples than I think any, and no disrespect. I'm sure your samples are great. So.
All right, that was a really good question and great answer. And you're all set? Okay, I really liked this session. I think we could do it every year, because this field is changing so fast, and that by next year we'll have a whole set of different topics to discuss at a similar session like this. If you want to put a suggestion on the suggestion board, you two could share a session and have fabulous speakers like this. How are we doing on time?
We got 12 minutes.
12 minutes?
Yeah, we got a lifetime. Yeah.
I guess my question is, you started out saying, oh, we had to do whole genome amplification. I know that's not the best, but then it ended up uncovering this thing that you wouldn't have seen if you hadn't been amplifying. So I wonder, do you think, would you want to try different polymerases that might be tolerant to different bases or modifications that you might then be able to see something that you also didn't know was there? Are you thinking about that?
Definitely, yes. That is one of the areas we're going to try to experiment into. Another avenue that we're working on is getting even more genomes and changing the diameter sizes. But I really hope that we can start to do more of the different polymerases. And I think with like if we could do it on even like bulk DNA too, using like Oxford Nanopore and just tweaking the base calling or something, that would be another way to see what else is out there. Thanks.
Actually, Alaina, can I follow up on just general questions? I'm wondering if you're avoiding co-cultures is also biasing the analysis. Like there may be organisms that only grow well together, and when you're like, I don't want doublets, you're also losing some important ecology there. And then second, can you do either cultivation in the trendy beads to like use microbial growth to then help you with de novo assembly? Because also always when you have one cell and you're doing whole genome amplification, it's hard to get, even if you've got a lot of whole genome amplified DNA, it's hard to close the genome still with short reads.
So to your first point about getting doublets or something, we are, in the ocean, we're targeting the free living plankton, so not particle associated, but that is another challenge that we're potentially missing those doublets that might be abundant depending on what other methods. And so yes, but we don't care if things are alive or dead. So if we can separate those doublets, we don't know if that cell is existing as a doublet or a pair, but we could still potentially sequence it as long as we're able to dilute it and treat it to the way that they separate. In terms of amplification, oh my gosh, I'm already forgetting.
Growing the cells.
Oh, growing the cells. Yes, so actually this method, I think, was initially designed for culturing. And so we've had people, their benchmarking was done on Bacillus, I think, and E. coli. And so, yeah, there's a lot of work going in there. I'm not on that side of things, but yes.
And then maybe one last one on, what about differential cell lysis based on what extraction method you're using? I'm just wondering if that's also a way you could enrich for some rare species if, I don't know, if the organisms you don't want don't lyse well under the extraction conditions or cell lysis conditions that you use.
Yeah, no. So our lab uses one approach, and I am on the bioinformatics side of things, so I am not very versed in which lysis they use, but there was a study with another partner of a trendy coming out of Sweden, but I forget which group it was, that tested different lysis chemical treatments, and they did yield slightly different communities, so there's a lot to play with this method. We wanted to find aliens, but we didn't find aliens. But maybe someone will.
One thing that we use in metagenomes when we do metagenomic sequencing is we use this enzyme from Sigma called metapolyzyme, which is a community of it's a mix of 6 different enzymes that are supposed to open the cell walls on the majority of the cells. And that enzyme was developed actually here at IBRF by a study group. That they say, okay, when you use these 6 enzymes, majority of the bacteria are targeted. So with that allows us to do a cell lysis and then a very, very gentle bead beating so that we preserve the long reads for long read sequencing. Yeah. Mm-hmm.
I have a question about the SAGs that you're generating. I would imagine I would have thought you'd want to separate out viral cells from bacterial cells based on genome size. Do you see any skew in representation based on genome size, and how do you handle that in that setup?
Yeah, so it's a complicated thing because some viruses have very large genomes, and our approach right now is looking at the percent of genes that have a cellular homolog based on eggnog database or whatever, and then we use a viral detection tool to look at similarity viral sequences. And so in terms of length and size, even if there's an infected cell with viruses, if that virus genome is small, you're still going to get a small assembly length. We've only saw a couple viral genomes that were on the end of 100 kb or so. So these are still very small assemblies. So size hasn't been a huge factor in being able to distinguish that right now. If we do some pretreatments and try to target larger particles, maybe we would start to see more blurred lines in virus cells versus virus particles and getting cellular genome together. Yeah. Did that answer your question?
To follow up on that, I was wondering if you have any experience with series nanoparticles, the hydrogels that are a capture and enrichment method. So what I'm thinking is you could use those as a bulk sort of enrichment from large volumes of seawater, avoiding your 300 microliter problem, right? So you take the concentrate from the hydrogel and then put that into your bubbles. You might get a better representation, but I don't know if the hydrogels work well enough to capture the full range of sizes that you'd like. I know it works well in sewage for viral sort of profiling. But
Yeah, we're very excited about seeing that method out there as well. Yeah, we haven't looked into doing any pre-treatment work, but there's definitely a lot of potential there. One kind of, I don't know if it's a limitation, but challenge is that we really dilute the heck out of our samples because we don't want co-occupancy. And so even if we have a very high concentrated sample, we're still going to dilute it to a 1% occupancy. So it is, yeah, that's still definitely a challenge there, but that would be a great way to just get rid of the cells at least.
Gonna switch gears here. So in regards to FiberSeq, I'd say like 90% of what I sequence on my Revio is FiberSeq. I'm doing none of the FiberSeq sample prep. So is that something you guys have internalized? And how are you guys going about like the optimization of that, of treatments? Like, you know, we've been doing it since pre-kit days. So I've always cautioned my people to do lots of optimization in terms of the HEA5 incubation. And I'm wondering if you're finding that as like a service that you've had to alter that.
Yeah, so I wanted to present FiberSeq. We haven't used it yet. So we are talking with the company, and it looks like there is optimization that needs to be done because they released the kit, but it's still, you know, you have to go on a sample by sample cases. But yeah, there is a lot of optimization that it needs to be done. Yeah.
So are you planning to offer the FiberSeq treatment service or just accept the treated DNA?
No, we plan to offer the full service. Yeah, we are working with Epicipher, trying to get it a little bit more mature, and because that's what we need. We need kids that are ready to for service. But we plan to offer the full service. I don't think people can deal with high molecular weight extraction, things like that. That's our expertise.
Yeah. Awesome. Thank you.
Mm-hmm.
All right, I think we have time for one more question. Anyone? Well, this has been a really cool talk. Lots of concepts covered. Go ahead.
I'll make one other. I had a discussion with Eric Chow last night about the N6, and one of the issues with the N6, because it's most often running with a cyber green double-stranded dye, that if you have non-specific amplification products or dimers, those will of course contribute to fluorescence that can terminate the reaction earlier than you might otherwise want. And he had this idea, and I'm like, why the hell don't I think of these things? He's like, why don't you? They're going to come out. N6 is coming out with a denaturation. What the hell is it called when you do qPCR? A melt curve analysis. So he's like, okay, do a melt curve analysis, find out where your primary peak of interest, the TM of that is, and then just do the fluorescence detection at that TM. And it's just like, God damn it. I'm like, you're a genius. I'm like, why am I not a genius? So I think, to me, that is really brilliant. So then we'll have another mechanism by which you can normalize for the product that you want, and then when you do size selection on the pool, you'll be able to have normalized libraries. So I think there's ways around the things that are maybe most problematic with the ICON PCR. So
That's the magic of ABRF. We have these after-hours conversations, and you get to rub elbows with the geniuses among us, and there's a lot of learning that goes on. So thank you again. This has been really great.
Thank you all, and like Barbara said, I want to do this again next year. So pitch me ideas, and we'll have a bigger panel. So thank you all.